In 8th and 9th class, we often use the microscope compared to other classes. So, we learn different type of the concept to the microscope. We can easily observe the slides under microscope if you know the how to read the microscope. I think you are all know about the parts of the microscope. Once again look at here, this is the eyepiece, this is the body tube, another name is barrel, and this is the revolving nose piece, object piece and stage, mirror and base. These are handle, fine adjustment, coarse adjustment. These are all, I think you are all very well about the these parts. You can easily use how to use these all of these parts. But whenever we are going to observe under a microscope, we may not see some objects not clearly. For example, we are going to watch from the object lens, there is no white field of lot of dust and smudges. Then we can't see the slide properly. That's why we should ready our microscope to watch slides clearly. That's why we must know how to clean the microscope. Now I am going to ask questions about the microscope. Look at here. Which problems may face while observing the microscope? The second one is, why does microscope has two type of mirrors? How do we use the fixation screw of the microscope? Next, how to clean the lenses of the microscope? So, if you know the answers of these questions, I think you are very well to handle the microscope. So, I would like to explain to how to clean and how to ready the microscope. Before going to use the microscope, ready the microscope. First you focus the light to the microscope. For this you use the mirror. Just you rotate the mirror until you watch the white field in the microscope. Watch it. So, here is not clear. Another thing is, so how to use a mirror? Look at it, look here. This mirror, right? Two type mirrors are concave mirror and the flat mirror. When do we use these? Flat mirror we use outside of the class or laboratory. In cause low light, we use the concave mirror. Now I am in the chamber, that's why now I am going to using the concave mirror. Once again I am fixing it. Now you watch through the object lens. Now I am observing lot of smudges and dust particles. Now how to clean the microscope? That's why I would like to remove the eyepiece. Then I am holding the microscope and tapping on my palm. I am tapping the microscope. If any dust particles and other things are in, they comes out. Okay. Now keeping once again. Now I am going for the eye lenses. It has two lenses. So it has the threads. Just I am keeping here. Now I am taking the little bit of the spirit. Just I am pouring the spirit in the small beaker. And now I am taking the earbuds and taking the little amount of the spirit. I am washing the lens thoroughly, upper side and lower side. Now I would like to wash these lenses with clean cloth. Now these any dust particles are on this lens, they will be removed. Now here is very clear. Now just I am keeping here. Once again reversing it, it has also another lens. It is also I am doing same thing. So I am removing easily, and once again I am applying the spirit on both sides. So now I am wiping it the clean cloth. So it is very clear, there is no any dust particle. Once again I am fixing these lenses to the eyepiece. Both sides these lenses I am fixing. Now object lens is ready to use. I am once again keeping here. Now just again I want to go to clean the object lenses. For this I am reviewing it. Sometimes it not come easily. So then I am using the cutting blade just to remove these object lens. So it has also threads. I am also removing the upper lid. So here is the spring. I am keeping here. I am removing the lenses. If lens is not comes out, so you can insert the spirit content earbud in the lens, and you clean the lens both sides. Once again you wash the outermost part of this lens. So wipe the lenses outside with the clean cloth. Once again you fix the object lens to these places. So this is fixed. Clean the other lens also similar way. So now you rotate it, keep the slide under the microscope, watch from here. Now the white field is very clear, so there is no any dust and smudges. Now you keep the slide and watch under microscope. Sometimes the white field is very clear, but I am unable to watch my section. Why? Think about it. Because my section is very thin. So that's why the light is dominating the section's barrier. So then we go for the diaphragm. Just you close the diaphragm. This is the diaphragm. When we are using this knob, it can close and moves. So it is a open very much. The light goes into these lenses. Then we are unable to see the section of the slide. So that's why then we gradually close this button towards a close position. Then the light is gradually slow down. Then we can easily observe the microscope. So this way we properly use the microscope. So when we will not face any difficulty while using the microscope. I think you are understand very well. You will be do like this. Thank you.